Hello students, welcome to Medico Apps Masterclass. Today I'll be discussing an important topic on food poisoning. Food poisoning is a term which you must have all heard about it, and some of you might have even experienced it. So what exactly is food poisoning? When I say this word food poisoning, so it is basically an illness which occurs in the body through a contaminated food or some drink. Now, this can be either through the microorganisms and their toxins or through the chemical poisoning that has contaminated the food or the drink you had it. But if I use a word strictly as the bacterial food poisoning, what do you understand by that? That means here the causative organism we know is the presence of bacteria in the food or drink, and that will lead to acute gastroenteritis. So, in this lecture, we'll be discussing about what exactly is food poisoning, its type, the foodborne botulism, which is also a type of food poisoning, the clinical features with which the patient will be present to us, and the diagnosis part. So firstly, as I have already started this lecture with the basic or the introduction of food poisoning, that it's an illness that is acquired through contaminated food or drink. Okay, and that is because of the either microorganism toxins or due to the chemical poisoning. And if I use a word bacterial food poisoning, that is due to the presence of bacteria in the food or drink that will lead to acute gastroenteritis. Now, this food poisoning can be of various types, but here we'll be discussing mainly the three types of food poisoning, and among them the first type is the infective type. Now, if I say that it is an infective food poisoning, that is due to the infective dose of microorganism is ingested by the person with the food, along with the food, and after the ingestion of the food, there is multiplication of this bacteria in vivo, and the incubation period is around 8 to 24 hours, and that food poisoning basically occurs due to the salmonella or campylobacter jejuni. Okay, so this is the first type where the infected dose of microorganism is injected by the person through the food, and the example is salmonella and campylobacter jejuni. Please do remember the examples here. The second type I am going to discuss here is the toxic type. Now here in toxic type, preformed bacterial toxin is ingested with the food. So already a food is placed, a food is there, and on that food some preformed bacterial toxins are formed. And here the food poisoning incubation period is 2 to 6 hours. And what will be the example? So the example will be staphylococcal food poisoning, the food poisoning due to Bacillus aureus or Clostridium botulinum. Now this staphylococcal and Clostridium botulinum is an example, is an important example of toxic type. And the third type is the intermediate type of food poisoning, where the bacteria is ingested with the food and then the bacteria releases the toxins in the gut. Okay, and here the incubation period is 6 to 12 hours, and the example is Clostridium perfringens food poisoning. So what was infective type? Infective type was when the infective dose of microorganism was ingested with the food. Toxic was when the preformed bacterial toxin is formed on the food and then it is digested. And the third is the intermediate, when the bacteria is ingested along with the food and the bacteria is then releasing the toxins in the gut. That is the intermediate type. And what are the examples? The infective type of example is Salmonella and Campylobacter jejuni. The toxic type is Staphylococcal, the Bacillus aureus and the Clostridium botulinum. And the last one is the intermediate, where Clostridium perfringens is the example. Now, in detail, if a short note is formed on the foodborne botulism. Now, what is this foodborne botulism? It's also a type of food poisoning, but it's a severe fatal form of food poisoning. Please do remember this, that if a short note is asked on foodborne botulism, It's a severe fatal form of food poisoning. And how it is formed? It is formed by ingestion of the preformed toxins in the food, which we have just discussed, where the food is contaminated with the Clostridium botulinum. That means it is a toxic type of food poisoning, and the incubation period is 1 to 2 days. Now, this food-borne botulism food poisoning is associated with the canned food. Now, there are various types of preformed toxins which are discovered, but among them, toxins type A, B, and E are the most frequently associated food poisoning toxins which are associated with the human disease. So, please do remember these important points in the food-borne botulism, where the food contaminated with the Clostridium botulism. Why? Because there is ingestion of the preformed toxins. The incubation period is 1 to 2 days, and it is basically associated with the canned food. And how would a patient present to us? So, a patient of food poisoning will be present with nausea, vomiting, and abdominal pain. That is a general feature with which the patient will be present. Along with that, in case of severe and fatal food poisoning, the patient might also be present with neurotoxin effects like the cranial nerve involvement, affecting the oculomotor muscles. And if the oculomotor muscles are affected, definitely there will be diplopia, drooping of the eyes with the squint. There will be vertigo, blurred vision, and dry mouth will be there. And lastly, there might also be progressive symmetric descending flaccid paralysis. Now, what is this flaccid paralysis? Flaccid paralysis is when there is shrinkage of the muscles, and due to the shrinkage there will be weakness, and if there will be weakness of the muscles, that might even affect the respiratory muscles, and if there is weakness of the respiratory muscles, that will lead to respiratory failure, and lastly the death of the patient. So these are the important clinical presentations with which the patient will be present to us. Now, coming to the diagnosis, how will you confirm the diagnosis? So for that, we take the specimen from either the food, the vomitus of the patient, or the feces of the patient, or either the blood. And what we'll do? So we'll use the smear made from the suspected food or feces, and will examine by gram staining. If it is gram positive, sporing bacilli can be seen, and culture may be done on blood agar or cooked meat broth under the anaerobic conditions, and detection of toxins in culture fluid is done by toxigenicity test in the mice. So this is how we confirm the diagnosis for the food poisoning. So I hope you have understood this lecture of food poisoning, where I have started with the basics of food poisoning, that it's due to the contamination of the food or drink, which can either occur due to microorganism toxins or either due to the bacterial toxins. Then we have discussed the three types: the infective, toxic, and the intermediate. And then we have also discussed in detail about the food-borne botulism, which is an important type of food poisoning with their clinical features. And lastly, to confirm the diagnosis, we will perform certain diagnostic tests, and the specimen will be used from the food, vomitus, or feces, or the blood from the patient. So this is food poisoning topic for you. Thank you.